In today's session at Medical Sciences, we want to discuss about Baltimore classification, or what we can call genomic classification of viruses. So as we begin, we always remind you to subscribe, you like the video, and share to others. And also we encourage you to join the membership. In this channel, we are asking you, if you love the good work we are doing to the society, you can support Medical Sciences by Naftali Muhumuza by subscribing every month to join the membership. So you are obliged to do that so that we can continue to serve the community. So about Baltimore classification of viruses, as we have seen, there are different ways. Sometimes the viruses are classified according to the shape of the capsid, or classified according to the presence or absence of the envelope, whereby some are enveloped, others are naked. Then according to the symmetry of the capsid which is the protective coat surrounding the genome, it can either be helical, they can be icosahedral. Icosahedral means they are 20-sided with 20 triangles, which are equilateral triangles, 20 equilateral triangles, whereby under helical we always have, like poliovirus is part of helical structure. Then others can be complex, like the bacteriophage. So these are different classifications, but in today's session we are aiming at the Baltimore classification. Baltimore classification, or what we are calling genomic classification of viruses according to the type of the genome or the nucleic acid. You can call it genome or nucleic acid present in the virus, not by shape or not by envelope, because we have seen by shape of the capsid can be helical structure, or they can be icosahedral, or they can be complex. Then according to the envelope, some are enveloped, others are naked. But in this session we are aiming at genomic classification, and they are different. According to Baltimore, there are only 7 classes of viruses, and they include, we shall have class 1. So there are 7 classes. He categorized them into 7 classes according to the genome. And the first one is, we shall have class 1, we shall have class 2, whereby we can begin with classifying DNA viruses, whereby for DNA, those ones with DNAs, you find some viruses have double-stranded DNA, that it becomes class number 1, and others have single-stranded DNA, and this one becomes class 2. This is class 1, this is class 2, and those which are double-stranded, for them, they have both positive and negative sense, meaning they are directly converted to mRNA. They are directly converted to messenger RNA so that they are transcribed, or they are used for protein synthesis. Whereas single-stranded DNA, these ones, they have single-stranded, these ones, they are also first converted to double-stranded DNA using DNA-dependent polymerase, and then later to messenger RNA as a template. And examples of viruses which are double-stranded, we have viruses like poxvirus, poxviride viruses. We have examples like hepadina viruses, or we can have herpes viruses. We have examples of adenoviruses, that is adenoviridae. So these belong to DNA strand, double-stranded. They are double-stranded. And even another one can be polyoma viruses or polyomaviridae. So these are examples of families which are double-stranded. And we have said these double-stranded, they have both positive and negative sense, that is the antisense, so they are directly converted to mRNA. But whereas the single-stranded DNA viruses, for them, they are first converted using DNA-dependent polymerase to double-strand, then later to messenger RNA, which is used as a template for protein synthesis. And examples under class 2 viruses, we shall find examples like, we only have one example, which is the parvoviridae. Parvoviruses. This is the only example which we have under single-stranded RNA. And these are not commonly, do not commonly affect human beings. They majorly affect insects and other animals. So they are not of much of clinical importance. Then classification number 4, we shall see that RNA viruses, those ones which have RNA, they can either be also double-stranded RNA. We can see some can be positive sense single-stranded RNA, and others can be negative sense single-stranded RNA. So this one will be class number 3. This is class number 4. This is class number 5. Whereby class number 3 is double-stranded RNA, which is class number 3. Then single-stranded RNA with the positive sense, that is number 4. Then single-stranded RNA with negative sense, that one becomes number 4. And examples of double-stranded RNA, meaning double-stranded RNA, they have both positive and negative sense, so they are directly also converted to messenger RNA for transcription or for formation of proteins like the capsid. An example here of double-stranded RNA, we have only retroviruses, which are also not much of medical importance because they affect majorly animals like insects and birds. So retroviruses are example of Baltimore classification class 3, which is a double-stranded RNA. After knowing double-stranded RNA, then we need to know about a single-stranded RNA positive sense. So class number 4 is becoming single-stranded RNA with the positive sense. And this because they have a positive sense, to replicate they are first converted to negative sense single-stranded RNA and then converted to messenger RNA using RNA-dependent polymerase. So we are seeing, because single-stranded positive sense, they lack the antisense strand or the template strand for transcription to form mRNA, they are first converted to negative sense, which is the antisense strand, then later converted to messenger RNA, because antisense strand is the one that is used as a template for formation of messenger RNA, which can be used for protein synthesis. And examples, here we have many examples of single-stranded RNA with a positive sense, and they include picornaviruses. We have picornaviruses belonging here. We have other examples like flaviridae or flaviviruses, or you can call it flaviridae. Then we have togaviruses. We have coronavirus, which we are calling COVID. Coronaviruses are also belong here. So these are the 4 examples of single-stranded RNA with the positive sense. We have picornaviruses, flaviviruses, toga viruses, and coronaviruses or coronaviridae. So these are the examples which need to first be converted to negative sense so that they can be transcribed into what we call messenger RNA. So you need to know the class and the example, and we are saying this is class number 4. Don't forget. Then class number 5. Class 5, we have single-stranded RNA. Single-stranded RNA with negative sense. So these ones with negative sense, for them, they are directly converted to messenger RNA since they have the antisense strand. So these ones are directly converted to messenger RNA for translation because they already have the antisense or the template strand which is required for protein synthesis. And this single-stranded, they are also much of medical importance, and examples of viruses under here include arenaviruses. We include arenaviruses. We include the Retroviruses. It includes orthomyoxaviruses. We find there bunya viruses, or what we call viridae viruses, and lastly paramyoxa viruses. So these are the 5 examples of single-stranded RNA with negative sense according to Baltimore classification, which for them directly they are converted into messenger RNA by the process of transcription so that this messenger RNA can be transcribed to form proteins. Then after seeing, now this is class 5, from class 5 we go to class 6, and this one shall give another smaller subclassification of retroviruses. And under retroviruses, we have 2 classes. We have only 2 classes, whereby we have class 6 and class 7. And class 6, these ones, we have single-stranded RNA with reverse transcriptase, and these ones use reverse transcriptase enzyme, whereby this reverse transcriptase enzyme reverses this single-stranded ribonucleic acid to form double-stranded DNA. And after forming this double-stranded DNA is the one that is converted into messenger RNA. And these ones examples majorly include the very infective virus known as HIV. So HIV belongs here. Human immune virus, immunodeficiency virus. Human immunodeficiency virus, which causes AIDS, belongs to this class of single-stranded RNA with reverse transcriptase. So they are reversed from single-stranded to double-stranded DNA or RNA. That is class 6. Then lastly, we look at class 7. And this class 7, we are finding they are double-stranded. This one includes double-stranded DNA with the reverse transcriptase. And an example here, this also uses reverse transcriptase. And an example here, we find hepatinavirus whereby a peculiar example is hepatitis B. So hepatitis B belongs to class 7, which is of retroviruses, which utilizes reverse transcriptase enzyme to manufacture their messenger RNA. So in summary, under Baltimore classification, we have seen 7 classes, whereby we have seen class 1 for double-stranded DNA. Then we have seen class 2, which is single-stranded DNA and examples, whereby an example is only parvoviruses. Then we have seen class 3, which is double-stranded RNA. They are also rare. We have only real viruses. Then we have gone ahead to look at class 4, which is single-stranded RNA with the positive sense, which are first converted to negative sense to be transcribed, to form a messenger RNA. We have seen examples there. Then we have seen class 5, which is single-stranded RNA with the negative sense. Then lastly, we have seen retroviruses, which has class 6 and 7, whereby class 6 have single-stranded RNA with reverse transcriptase enzyme. Then class 7 double-stranded DNA reverse transcriptase, where we have seen hepatitis B being a member. Thank you so much for learning with us. May God Lord bless you.